Good morning. It's 9 o'clock here in San Diego, so we're going to start the webinar. The title of today's webinar is Ligand Design Using the ICM Interactive Ligand Editor. My name is Andrew Ori and I'm a Senior Research Scientist here at MolSoft. So if you have any questions after the webinar, please email me or call me. During the webinar, please use the chat or text messaging option in the GoTo meeting panel that you have on your screen. If you're interested in following MolSoft, we have a Facebook page and LinkedIn, Twitter, and also on YouTube we have a lot of tutorials and webinars on YouTube. So today we are going to focus on the Ligand Editor. The editor is widely used throughout the pharmaceutical and biotech industry for ligand design and optimization. The tool was built in close collaboration with medicinal chemists at Novartis. It's a fully interactive tool whereby you can make changes to a ligand in a 3D environment and see the effect of the change on binding, or you can make changes to the ligand in the receptor using the 2D panel on the left shown here. The story with Novartis was published recently in an ACS journal, and part of that paper describes the Ligand Editor. So today's topics, we're going to look at ligand receptor visualization, so you can see the pocket and where you can make changes. We'll look at ligand editing in 2D and 3D, how to evaluate the effect of a change on a ligand, look at the capabilities of the ligand editor, so you can fully undo everything all the changes you made to your ligand and go back to the original pose. And then if you make a change and you like that change, you can save that change directly to a chemical spreadsheet. We got look at ligand docking and the minimization and refinement of the modifications that you make. I'll show you how to use distance restraints and tethers during docking. You can also screen a database of substituents to a particular point on a ligand. And there's also embedded in the ligand editor, there's sort of more advanced docking options such as covalent docking and fragment docking and linking. Methods to incorporate induced fit using multiple receptor conformations, and also if you don't have a protein receptor, you can also design ligands in 3D pharmacophores inside the ligand editor environment. So we're going to focus on one structure today, and this structure is riscovitine. It's quite an interesting inhibitor, a kinase inhibitor, because it's currently in clinical trials for quite a few different diseases, shown here, and most recently cystic fibrosis. It's an experimental drug developed by a British company in Scotland, and I think they also have a company on the East Coast in the USA. Preferentially inhibits multiple enzyme targets, including CDK2, 7, and 9. It alters the growth phase or state within the cell cycle. And there is a nice crystal structure with this molecule bound to the ATP binding site of the kinase. So we're going to retrieve this molecule and set it up in the environment. 1 UNL, and the ligand editor and the ligand editor panel here. So we have 1 UNL. This is the ligand. We go to the button here, and it asks us which is the ligand molecule. There's only one drug in this protein structure, and it has this ICM command line definition of the ligand. You can also assign charges based on the pH, and we go OK. So now you should be now the ligand is set up. The next step is to set up the receptor. So we go to the receptor button here. The receptor object is 1-UNL, which is this structure. Then we have some options. We can keep tight waters that make 3 or more hydrogen bonds with the ligand or the receptor, with the ligand and the receptor. We can keep all the waters in the pocket, or we can delete all waters. And then, because it's a PDB structure, we need to optimize. We need to add. We will add the hydrogens, and so we need to make sure that we also optimize the hydrogen bonding network. And so we optimize asparagines, glutamines, histidines, proteins, and you can choose to mask missing side chains. So if the crystallographer hasn't seen atoms on certain residues, these residues will be masked out. If you uncheck this, ICM will build any missing atoms of residues in the pocket. So it's a preference that you can choose. So in this example, and in most cases with a ligand editor, you have a pre-existing structure, and the ligand is bound to it, so you can choose make box around existing ligand. This is the energy box, the box where we build the energy maps, and you can make it bigger or smaller by dragging on the corners of the box. Let's go OK. So this builds the maps. So once the maps are built, you have the complex with the ligand and the receptor. So we now need to start visualizing, where we can start building and improving our ligand. So there are different options for doing that. We have option to display the receptor surface colored by binding property. There's also an alternative surface, which is a sort of combination of the receptor and the ligand, called the ligand pocket surface, which gives a better representation of where you may have cavities in which to build. This I will show you in the ICM. You can display hydrogen bonds. You can display energy circles, shown here. So for example, if something's making a clash, you have these big red circles which represent poor energy, whereas the rest of this ligand is okay, but it's obviously making a clash to the receptor here. You can display the relaxed ligand compared to the docked ligand. So this is quite important, because when you are when you're designing new ligand, you may be building in strain to the molecule, and so you can see how strained. We have an energy calculation, but you can also see how different your design molecule is compared to the relaxed version outside of the pocket. You can also display unsatisfied hydrogen bonds, so it gives you an idea where you might be able to build towards in the receptor. Okay, so practically in ICM to do that, we set it up. We set up the ligand and the receptor, and then we have some options here to display. So we can drop down here, the receptor pocket, shown here. So white coloring, the pocket is displayed in the ICM workspace here, and you can change the representation. So it's going to make it, at the moment it's transparent, that's going to make it solid smooth. So white is neutral, green is hydrophobic, blue is hydrogen bond donor on the receptor, and the red is hydrogen bond acceptor, and green is hydrophobic. So that's one representation. You could also display the pocket surface, the ligand pocket surface, which is a slightly different representation. So it's actually colored the opposite way to the receptor pocket surface. So this is a red is telling you that you need to add a hydrogen bond acceptor into this region to make a, to interact with a hydrogen bond donor on the receptor. And it's also very useful because you can see that there's obviously a cavity here that could be built into, but you can see here that this region is completely closed off, so there's no way that you could add another substituent to this atom without making a clash with the receptor. So it's a useful way of viewing it. Okay, so you can also display the hydrogen bonds. There's a button here for hydrogen bonds, and we can see that this kinase inhibitor makes two nice hydrogen bonds with the hinge region, which is a common thing for kinase inhibitors in the ATP binding pocket. So just get some. So we have the hinge region here, the glycine-rich loop here, and the N-terminal and a C-terminal down here. Just going to undisplay that. So the hydrogen bonds are color-coded by how the angle and the distance. So a thick green hydrogen bond is a strong hydrogen bond. You can evaluate, you can display the energy circles. So everything is fine here. We've got green, but low values for each of the atoms. You can display unsatisfied hydrogen bonds. So we've got two here, one here, and one here. When we start making modifications to the ligand, you can display the relaxed ligand. You can display our stereo labels, and we also have an inbuilt option called Pretty View, which changes the view. You can modify that to how you like it. And here on the right-hand side is the button that calculates the score for this ligand. So this is a VLS score, and this is the score we'll be concentrating on. It's so the lower this score, the better the prediction of binding for that ligand is. You can toggle the hydrogen display. So usually a good one for, you can show all hydrogens, or ligand all hydrogens, and so when we start modification, you'll see that we need to select the ligand. Okay. So you can, as I mentioned, you can edit in the 3D environment, or you can edit in the 2D environment. In the 3D environment, you can change atom types, bond types, change the charge and torsion angles, and also select different substituents. So you click here, and then you click on your molecule, then you evaluate the score. If you don't like the score, you don't like the change, you can undo and redo. Same in the 2D environment. In 2D, you can just display the molecule in the 2D editor panel here, make changes using, it's like typical ChemDraw stuff. So you got bonds and atoms here, templates here we showed a couple of weeks ago in the webinar. And you can make changes, and then that's transferred to the ligand in 3D, and then you evaluate the score. So it's a pretty feature-rich sketcher. You can monitor the properties and other things. So once you've made a modification, you can save your modification in the protein form, with the ligand and the receptor in PDB format. What's the most common way is you make a modification, you like it, you're not sure if it's 100% what you want, but you want to go back to it, so you can just store it 3D coordinates in a chemical spreadsheet, or you can write it out into SDF, MOL2 format. You can export your chemical spreadsheet to Excel or HTML, or you can save the whole thing in a binary format, and you can share this with your colleagues who can view it in the free ICM browser, display on the web using a free active ICM plugin, or you can share on iPhone or iPad using iMolView, which is our mobile version of ICM. So once you've added it to a chemical spreadsheet, you also have the tools in the spreadsheet, such as chemical searching, editing, clustering, plotting, etc., which we showed 2 weeks ago. Okay, so we go back to ICM. All right, so let's start to modify this ligand. So I'm just going to remove these unsatisfied hydrogen bonds. So the structure is shown here. If we want to start modifying it, we click on this button here, which has a pencil and an atom, so it means you can modify it. And you have this panel of substituents. We have a panel of atoms. So for example, we can click on carbon atom here. We can change this oxygen to a carbon. You see the carbons there. We can evaluate the score. It won't like it. The score has actually got higher because we've lost that hydrogen bond here. That's okay. We can undo it and go back to our original ligand, and we've got the hydroxyl here. We can change bond types by clicking on a bond. And so, for example, we can change this to a double bond here. If we just click on the bond, we see now have double bond, and go back to a single, like this. So you can evaluate the score, and then you go back to a single by clicking on the single bond. Or undo. I can't. There we go. So it's gone back to there. Okay. So we can change torsions, which I'll show, but I don't want to do that just yet. We can change the chirality, the stereo, switch between R and S. We can erase atoms. Just click on the erase button, and if we don't like this one, just undelete. Evaluate the score again. So as we'd expect, it's gone up. It's got worse, and we can undo and put it back. Okay, so you see on the left-hand side we also have this panel of substituents. So let's go back to that view of the pocket, and it's called ligand pocket surface. And we can see it's over on this side of the, let's display the hydrogen bond. So we got the, it's upside down. So the N terminal is up here and the C terminal is down here, and we've got these hydrogen bonds here to the hinge region here. And the ATP pocket's here, the glycine-rich loop comes over here, DFG will be down here. So we can see that, in actual fact, we've got two regions here and here where we have some cavity in which we can build into. So I'm going to try and exploit, there's a bit of a hydrophobic region here that we could try and build into. Also some, it's telling us that you need to add a hydrogen bond acceptor here to interact with the hydrogen bond donor. So what I'll do is I'll select a few substituents. I'm going to select a couple of halogens and sulfonamide, a sulfonyl, and a couple of others. So you can see we selected 1, 2, 3, 4, 5, 6 substituents. And now we can screen those substituents onto this point, onto this hydrogen atom. So just click on that hydrogen atom, and you'll see that it says here trying these groups. And so it's basically docking these groups to the receptor. It's pretty quick. So once it's done that, it shows you a table, and this sulfonyl group has a much improved score of -40 for the VLS score. It shows the strain, so you want to keep the strain as low as possible. There's also prediction for log P and solubility and other properties. So this has a pretty good score, so we can just double click on that, and you see that the structure, it's made a hydrogen bond here between these two atoms. So it prefers that one. The next one in the list is a sulfonamide, and you can see if you double click, it likes it. It's a good -37. And then as a halogen actually improves it as well. Bromine. Then our original structure is actually, so all of those substituents that I chose improved the VLS score pretty dramatically from -33 to the top one, which was -40. So I like this sulfonamide one, for example. So I can add that, and I can put that into my chemical spreadsheet. So you click on this add current ligand to table, And so this is our ligand. This is our collection of structures that we like. So we can add more to this table when we want to. We can display it in 3D by double clicking. This table can be any size. You can add as many as you like to that table. Okay, so you can, as I mentioned, you can edit in 3D, but you can also edit the ligand here in 2D. It's by clicking on this 2D button here. So, and in this panel here, we have the properties of this structure. So we have the typical Lipinski rules, as we showed last time, and some other predictions for whether it's a pane structure, a pan assay, interference structure, the volume. Everything seems fine, but maybe we don't quite like the sulfonamide. Maybe we'll try something a little bit different, just to see if it binds. Because you can see here that there's a hydrogen bond accept. We need to put a hydrogen bond acceptor next to this hydrogen bond donor. So maybe we don't need the full sulfonamide group to do that. So we could edit it by use the eraser button, just edit this sulfonamide group, and we could add a carbonyl here and a carbon. So once I do that, we can commit this to 3D by clicking on this button here, and you can see that when we do that, we get the hydrogen bond. We also get the score of -39, which is pretty good, better than the original ligand. So bear in mind these are just predictions of binding, so you need to be careful what you're doing. But we also need to keep an eye on the ligand strain, and we're not making artifacts and molecules that would never energetically exist like that. But as we're doing it, we can see the score, the energies are okay. So for example, if I had made a big modification to a region where there's no space, or the molecule would have to strain in order to incorporate that substituent. So for example, here, if I click here, if I add this rather large molecule substituent, you see that it flags that there's an obvious clash. It doesn't like this substituent. That's okay, we can undo it and go back to our ketone addition we added, and I like this one, so I'm going to add this to our chemical spreadsheet again. And if you have a series of structures, you can actually tag them as well. So I'm adding it to a spreadsheet, but you may want to tag it, if it's, I don't know, series 1 or something, and then keep, so that will add a tag here, so you can sort this table by different values. Okay, so we made the modifications, but some substituent, we made very small modifications. So ICM does a very small minimization, and so you don't really, with small modifications, you don't really need to redock the whole structure. And yes, so you don't have to redock. But if you're making a very large change, you may want to redock it. So let's have a quick slide on docking in ICM from beginning. So the docking uses MolSoft's ICM docking technology, which has been around for, the original method was developed by Ruben Abagayan and Max Totroff back in '94. So it's been very well validated. It performs very well compared to other commercial programs. You can dock a modified ligand. You can dock, if you have an SDF file, you can dock a chemical spreadsheet to the pocket in the ligand editor. And you can screen a database of substituents, which I'll show. induce fit docking using multiple receptor conformations, another example. And you can also refine the side chains after docking or during docking. And there's covalent docking and APF docking, which is a 3D Pharmacophore method of docking. So in this example, we have, I'm just going to use what we call pretty view, which some people don't think is very pretty, but you can change that to however way you want it. So I'm just going to color the ligand green just for clarity, and color the carbons green. So I'm just going to undisplay the ribbon in this. So we can see if we redot this molecule, we know that this is going to make two hydrogen bonds with the hinge region here. So we may want to keep these two atoms at this distance. So we can use distance restraints to do that. And you click on distance button, distance restraint button here, and then we select the two atoms that we want to keep at the same distance. So it's these two, and you get this table where you can edit the maximum distance. You can make it a bit bigger if you wanted a bit more flexibility, and the minimum distance. There's certain weightings you can use. And then you can choose to use this distance restraint or not when you dock, and it's just to display this here. Okay. So when we redock this molecule, we're going to keep this distance here. You can also use tethers, which is useful as well. So we can say we want to tether this atom to this point, and it won't move during docking. So you can see when you click on the tether button, you get this red cross through the middle, and this atom is tethered to this point. Okay. So as I mentioned, so you can tether, you see this red cross here, and you can toggle it on and off, or use it or not use it as you desire. Okay, so to dock, we're in the Ligand Editor tab, and you can just single click on this docking button here. Okay. So this uses the ICM docking method. It uses it's a biased probability Monte Carlo method, and so it's randomly sampling the torsion angles of the ligand inside the pocket, and it's going to evaluate the energy and build up a stack of energetically favorable ligands, and so energetically favorable poses for this ligand, and soon it will provide a stack of confirmation. So you can, I've turned it off, but you can actually display the docking as it's going along, but this takes a little bit longer to do. Okay, so we've redocked, and we see that these two atoms are still in position, and it wasn't a great lot of changes. We've got an additional interaction here with this asparagine, showing here, and we kept this hydrogen bond which we made earlier. So you can see that the VLS score is -40. So you can toggle these, confirmate the different poses for each one using this box here. But the most energetically favorable one is the first one in the list, and it's got a score of -40. Okay, so during docking, you can add tethers and distance restraints. You can also allow certain residues to be flexible during docking. So you need to be careful how many you choose. You can, to do that, you just need to select the residues that you want to be flexible, and then go to Advanced option, and it says Define Flexible Receptor Groups. So in this example, I just chose one residue. You can choose to have the full side chain to be flexible, or you can just choose, if it's got hydroxy, you can just allow the hydroxy and water to be flexible during docking. Okay. Just for speed, I'm not going to show that, but you can, and this is pretty well-defined pocket, so we're not going to see much rotation of the ligands in the pocket of the side chains. But it is something to look at if you found a good substitution that you like and you want to double check that it's a good interaction. You can allow certain residues to be fully flexible. Okay, so as I mentioned before, you can also rotate torsion angles in your ligand by clicking on this rotation torsion button, and just let's rotate. We just so basically, to do that, you click on the rotation button here, and then click on the bond, and that allows you to rotate that manually to a different position. You can also manually rotate the side chains of the receptor, but you need to, if you do that, you need to remember to remake the receptor and remake the maps to accommodate the changes that you made to the receptor. So let's go back to the ligand pocket surface again. So as I mentioned earlier, there's two positions where we can modify. Okay, so using my orientation. There we go. So the hinge is here. So there was this region here at the bottom. There's also a gap here. So we can maybe we want to sort of experiment and see what substituents could also fit into this region here. This is getting towards the DFG region, sort of extending out into the allosteric pocket of the kinase, away from the hinge and away from the direct ATP binding site. So we could experiment with some substituents. So to do that, practically we click on an atom that we want to try. We go to Advanced, and then there's Find Group. So it's asking you, do you want ICM to calculate the full binding score for the full ligand, or do you want to just calculate a score for the substituent? If you're screening many hundreds of substituents, this will obviously be quicker. This is a bit slower because you're evaluating the full binding score. In ICM, we have a set of system modifiers, but you can also use your own table. So you'd have to read that table, put it as an SDF file, or you can sketch the substituents using the sketcher here. Okay, so once you do that, let me just delete them. Let's have an example with that result. So we go once you do that, and similar to the virtual ligand screening, basically you are screening substituents here. So we get like a hit list table, and we screened 45 different substituents. So the best one is this ring. I'm just going to undisplay that. So we can display it by double clicking. It's got the best score, but maybe this amide is yeah, so this is where we added it here. So you can see the ring, the amide has a good score, -37. I like that one, so I'm going to add that to our spreadsheet. There's a variety of other ones we can toggle through for each substituent. And actually, I think in row 17, or even further down, yeah, row 23 is our original ligand for scovidine. So we actually found 22 substituents which might improve this molecule. So we had a good question about synthesizability. So obviously you can carry on making your great ideas computationally, but how well that's can be synthesized is a different matter. So we don't actually have a score for that, but you can look at substituent. You can build a substituent database using reactions. So that can guide you as to which substituents are sensible for the reaction that you're using. So yeah, that would be useful to have a score for that kind of thing. But at the moment we don't have that. But you can use you can use our tool in chemistry. There's you can build a combinatorial library using a reaction, basically, which I showed last week. Okay. So these are the editing tools, and this is a table that you can sort by score here. I'm just using the sort button here. It's also a chemical spreadsheet, so you can make plots of things, as we showed last week or a couple of weeks ago with like this, and so yeah, so it's fully interactive. So you can evaluate which substituents are useful and which are not. Okay, so we showed for flexibility you can define flexible receptor groups. You can also undertake a receptor side chain refinement. So you can make a substitution, and then you can allow, for example, serine, or just serine, all side chains. So you can see the side chains are being refined with the ligand inside. This takes a little bit of time. But over the years of development ICM for flexible receptor, we found that the most efficient method is for flexibility is to use multiple receptor conformations. And it's easy to say that, and then if you have a lot of crystal structures and you're in an ideal solution, yes, scenario, you probably can see in your ensemble of crystal structures which parts are flexible, which parts are not. So you can use an ensemble of crystal structures to dock to, which I have an example. I'm just going to stop this refinement. It takes a little bit long. Okay, so I will send a paper that describes the multiple receptor conformation method when I send the video of this as an email, but just quit. So multiple receptor conformations, if we call it 4D docking, you superimpose all your structures and you dock to them and here. So in this example I have 3 nuclear receptors. That's the wrong one. And these are the same proteins, but the pocket differs quite significantly. So we have 3 similar that are identical from the same nuclear receptor family, same protein actually, but depending on the ligand and the pocket state, the pockets change quite dramatically. So we used the ICM. I used the ICM pocket finder to show the pocket. The pocket is shown in blue. So so what you need to do is so in this example we're lucky we have 3 crystal structures, but you may want to, if you only have one, you might want to use multiple receptor conformations of a loop region, for example, that goes into the pocket. And you can do that using the modeling tools in ICM. So to do this we need to superimpose the pockets. So to superimpose, we select the molecules we want to superimpose, and then we go to display panel, and there's a handy button here, the superimpose button, which will superimpose all those three structures on top of each other. It's a very useful tool because it reacts to whatever you select, it will superimpose. So if you superimpose this loop, it will just superimpose that region. But important to us in this example is that we superimpose the pockets here. So we select that region, go OK. The pocket is superimposed. So now, in a similar way as before, we go to Ligand Editor tab. I actually have a ligand here that I'm going to dock to these three receptors. So we click on this button. The ligand is 1-ACO. I found it straight away. It's going to undisplay the pockets just for clarity, like this. So now we go to the receptor setup. And the only thing that's different here is that rather than selecting one receptor, I selected all three of the receptors that I want to use in the docking. So I'm going to dock. So we're going to build maps for each of these three receptors. And it's similar as before. You can keep orders or not. Optimize hydrogen, asparaginase and glutamines. So it's going to make box around existing pocket. And go OK. So this is generating the maps for each of those three receptors. And so you can dock and make modifications in those three on the fly and see directly which one is which receptor conformation is best for your ligand. So you can obviously include more than three structures. You can include as many as you want, but you've got to have a trade-off with the if you have more than 5 or 6, you need to increase the length of the docking simulation to do this. So we can dock this ligand in multiple. But before, for single conformation, you just click once. But if you want to do it in multiple receptors, you click and dock, click and hold, and there's a 4D option shown here. I've actually, in this example, I've already docked it in the 4D, so I can evaluate the score for each one. So this is the third receptor. So the score is very bad, and we can see the why, because we have a lot of clashes in this region. So this is obviously not the best pocket for this ligand. We go to the second example. The score is still bad. It's a positive score, and we got not quite as many clashes, but we have 3, 4. And then in the first protein in this example, we have a great score of -30, and if we look at the energy circles, we don't have any clashes. So we can make modifications to this ligand in the 4D maps in the same way as I showed earlier with this panel here. Okay. So the final example is an example whereby you don't necessarily have a crystal structure, but you have a series of compounds or chemicals that you know bind very well, and you can, instead of editing the ligand in the pocket, you're editing the ligand in these atomic property fields, which is a method developed by Max Topchoff here at MolSoft, and it's a 3D pharmacophoric method. And so basically, in a similar way as before, we go to Ligand Editor. We click on this button to choose the ligand. I have a ligand in this example that I want to optimize. And go OK. So these are these surfaces are the property fields, the 3D pharmacophores. So in this example, we only have a very lipophilic region that's conserved, and these 2 hydrogen bond acceptor regions here and here. these are actually COX cyclooxygenase 2 inhibitors that we looked at a couple of weeks ago. And so I've superimposed these, and now so we go to the Setup Receptor button, and there's a tab, there's Protein, and there's a second tab called APF, which is the APF fields. This is ligand based, and so we're going to use, so we have 4 chemicals, 1, 4, these in our fields, and we choose selection of 4. Make box around these fields. Okay, so now we can just edit the ligand in a similar way as before. We evaluate the score. We can minimize the ligand in the field, so that improves the strain in the score. And then so we have 2 buttons, minimize button and docking. So it minimizes a shorter thing. So we can add a group here and evaluate the score. So in the same way we had these energy circles. It doesn't quite like that one. The score got a little bit worse, but it's a reasonable modification. You can undo it if you don't like it as before in the same way. Okay, so we covered most things. The other 2 examples are fragment screening. So in our original example, 1U and L, fragment linking. We have a fragment screening method in the docking menu, which is a different thing altogether. So if we imagine a scenario where we can just trying to make a fragment or something. Delete some atom. Okay, we have 2 fragments, and so we can then, inbuilt in ICM, there's a linker database of linkers, and so to link them, we just need to, we go to the advanced menu, and there's an option called Find Linker, and we have an inbuilt database that you can use, and then it will screen a set of linkers, and you can set the strain cutoff. You can set the maximum number of rotatable bonds in the linker, and then there's a cutoff for the contact with the receptor, and you go OK, and you'll get in a similar way as before with the substituent screen. You get a table of linkers for this structure. Okay, so that's the end of the webinar. The next webinar will be in a couple of weeks' time, August the 6th, and we'll be looking at molecular graphics in ICM, making slides and movies, importing those movies to PowerPoint. There's movies, and there's a fully interactive ICM inside PowerPoint where you can rotate the molecule in the PowerPoint and in HTML for presentations. Thank you very much for joining the webinar. I'll be here for a little bit longer to answer questions, and hopefully see you in 2 weeks' time.